My risk area is including the three area is the nanomaterials and the nanomedicine and the theranostics. I want to do the main focus on the cancer precision phototherapies. So we can use this material for the molecular imaging and get some smarter drug delivery systems, and also get some activation these immunologies. So as we know, these nanomedicines have a different function. They can loading such as the imaging molecule and loading the drug molecule together too. So we can combination these two, such as we can combination this diagnosis and the therapy together to a way to the precision nanomedicines. This is some commercialized nanomedicines such as Doxil or aberracines. So I also introduced some what is the phototherapy. The phototherapy is use the laser to excitations the photosensitizer, and then to excitation like RS2, and also the local higher temperature to kill the cancer cell. So they introduced the cell death and resulting this tissue necrosis and immunoactivations. But by now, the commercialized these small molecules, the PDT, these molecules such as the phosphorine and the phoscan, they have some different defects, such as they have a short circulation, easy bleach and non-targeting, and the low efficiencies. So we can use the different nanomaterial as a drug delivery system to get us these nanoprobes. So I use this indocyanine, the green is ICG nanoprobes to do the early diagnosis. Like we can use these nanoprobes to get the breast cancer in the very early the step. This is the very small one, is only 1 millimeter. We can see this one, and also we can use this technique to detect the cancer marginations. So you can very accurate to shining the lasers on these areas. So our first work is start on the 2000, and we publish our papers on the ACIS Nano 2013. In these works, we combining these chemicals agency, this Doxil, and also this ICG together in the lipsome systems. So we can see this very simple systems, but the therapy effects is very higher. You can see we only use the very low toxic dose, and only the 1/3 the normal dose. But normal dose is 2 milligrams per kilogram. So you can see after laser shinings, you can get the local temperature to the 15 degrees, and then result in the tissues damages, and then get the very good and the tumor ablations. And then we improved our this drug system, used human serum albumin, use the HSA as a drug delivery systems. You can see in this scene we get a very long circulating time is about to the 1 week is 7 days, and also the targeting is quite good. It's about the 35 times and compared to the normal tissues, and they can also be and get the PDT and the PDT together. Use these systems also to get a quite good these therapy systems. So for the cancers, these therapies, the first program is hypoxia, because we know in the tumor, at the center of the tumor, all is hypoxia. The hypoxia will be overexpressed as HIF1 and the result MD1 and also PGP and finally get the drug resistance. So we firstly introducing our work is the nano artificial red cells. So it is the oxygen nanocarriers. So we use the hemoglobins and use the ICGs and its complexes. At this condition, the oxygen will be transferred to the ICG to get the very higher, like this very high RS and efficiency, and get the quite good the SPDT efficient the therapies. And then we get use the proteins hybridizations to combine the hemoglobin and HS together. You can see is the hybridized proteins nanoparticles, and use particle you can get the very high and the down regulation that is HRF and the MDR1 and get the quite good the protein hybrid nanoparticles. And the second question is for the targetings. How we can get to targeting? But normally we use the antibodies and some such as other things or the small molecule like the folic acid and also the other things to get this targeting. But by now we introduce the different methods. Use the cancer cell membranes. This cancer cell membrane is just the from the cancer cell, extract this cancer cells membrane and then uses the membrane self-assembly or nanoparticle together to get the biomimetic nanoparticles. At this condition, they have quite good effects like the homologous-targeting. That means we have a different adhesion factions, so we can recognize such as we use MCF membranes, these nanoparticles can be only targeting the MCF tumors, and you can discriminations the different tumor type and also the normal tissues and get the good accumulations in the cancers. And use this method, we also get the HB together and get the very higher risk. Such as in our system, we have a good hypoxia, reduce the hypoxia, and go to the higher dose the loadings and get this oxygen enhanced chemotherapies. The third question is how we can improve the immune activations in the cancers, because for the cancer therapies, finally we need to use the immunosystems to use this to fight all these cancers. So we use the different cancer cell membrane, such as use the oxygen, the PDT, we make this dining the tumor cell, and then use the introduce the immunogenetic cell death, this ICD process, and these 3 marks to can be mature the DC and then also improve our CD4 and CD8 and NK the cells, and then introduce our activations. It is the immunities. And use this method, we not only can be kill the primary tumors and also some distant tumor, and also is effective for the anti-metastatics. So this is our result. You can see for the primary tumor, we can very ablation and suppress this the tumor growth, and also for the distance they have very quite good suppresses. And for the metastasis, these cancers we can see is disappear after our treatment. So that means use this method, we not only use the PTT to kill the cancer, and also we use this activated immunity to finally to get the tumor ablations. And this is use the NK membranes. It is another way this is for the normally for the ICD process. They also have another way for the M1 macrophage polarizations to activation the another more the immunity behaviors. This is some analysis. We use the proteomics this analysis to analyze these 868 proteins and find some very interesting proteins, such as this for the targetings. And this someone is figure E is showed some targeting protein and also M1 polarizations. List some the evidence for such as for the CTR, ATP, and HMGB1. This marked introductions, and also some mature CD4, CD8, and also some secret factor, the TNF-alpha. All these conditions we can see it is a very good one for the tumor by these immunoactivations. And we also use the macrophage cell membrane mimicking this nanoparticle to transfer to the glioblastoma, and we use the VCAM1, these passengers, and use this one. We open the BBB and get the drug delivered to get to the direct to this cancer cell, and you can see to get also get quite good and delivers efficiencies. And another way we want to modifications the T cell, because you know the CAR T is a very efficiencies for the is a good method for the cancer therapies. So can see we use the clinical chemistry, and this is the isos sugars to the metabolic labeling this T cell, and also use this membrane together on this. We use the nano-bio-inspiring and immunodegradations and clinical targeting, and finally get the photothermal PTT to kill the therapy. This is only use the T cell membrane. We also use this CAR T engineering for the cell immunotherapies like this. We use the BCN, these BCN sugars, and also the iso sugars, this clinical chemistry. So our T cell and also tumor cell can be recognized. This is like artificial targetings. We introduced our artificial targets on the T cell and the tumor cell, and they can be recognized together. And use this one, we use the larger cell as our models. It can be very higher efficiency for the blood tumor and also for the solid tumor. For this one, we find this method can be used as living drugs, such as the cell is a drug, and can be very high efficiencies to kill the cancers. So next, because we know for the nanoparticles, we have a different formulations. So for the controllable, is something is not well observations. So at this condition, we want to make all these characters to the one single molecule. Like I said, we use the donor and accept these design principles to get and also use the near infrared molecular images, like the first windows and the second windows, to get the very clear, very higher clear list near images. So use this method, you can see this Theranostics, single molecular probes. So we can they can switch on by the laser and can be seen quite good and also responsible for the pH. For the time schedules, I also introduced this molecule. This molecule is for the hypoxia response of the near to these probes. You can see this near to images and the photoacoustic imaging, also PTT, is also on the hypoxia these systems. Because for the hypoxia, they have all expressed natural reductase. Use this one, we can transfer our nitrogen to the amine groups. So at this condition, we can, this spectroscopy is a huge change like this. You can be seen only one hydrogen change, you get the very 400 nanometers this movement, and get this one very higher clear, and you can see this image is very clear. There is no noise at all, and also the TBR is tumor to the background. The ratio is very high, to the 30. So you can compare the different the work. Is normally is only the 5 or the 14. So at these conditions, we use this method to get the quite good. And also, you can see the PTT behavior is also the very high efficiency in there. So, because for the these therapies, in spite of this, the photosensitizer, all these nano material systems, we also need these devices, is laser devices for the cancer theranostics. So use this instrument that you can get it together for the fluorescence imagings and also the photothermal therapies. And you can see this is a in vivo systems. You can see these are mice. This is the bright area is a tumor. After the shining the laser, the temperature increases. Is like bubbles. It's shining the where is the indicated where is the cancers, and we can also the tracing all the system, all these processes like the and see the how the tumor is killed and the tumor the conditions, and also we can see that this is a fluorescence, this is a thermal therapies. So the next step, we want to introduce this system to the clinical applications. Use endoscopes like this. We use optical fibers to the tumor size, and then shining this one and get the very higher efficiency one. And we have get some preclinical tested in the systems like this for the gastric cancers and also the brain gliomas and also head and neck cancers. And the left is show another clinical trial in the USA. It used another system. They used the gold shell, the silicon nanoparticles. At this condition, they use they also can be get the higher temperature in there, but this system is not degradable. So our system can be fully degradable. Is more safety and more efficiencies compared compare these systems. And finally, I'll introduce the new researches on the Nano-Biorobotics. This is like the self-driven therapies. Like this, we have a macro robot. This is algaes. We can combine some magnetic particles in this, and our photosensitizers in these bacteria systems, and use our near laser and also the hemorrhoid cores. These are the magnetical drivings. So we use the magnetic for field for the driving of these macro robots, and also use the NIR laser to trigger our system. So we can control the very clear controllable these systems, and also the shiny systems get finally quite good these curables. So finally, I want to see we need applications of our technique to the clinical applications. For the clinical translation, so we need the system very simplicity and the safety and the controllable and the large scale manufacturers, and also have a very good production pipeline and marketings. And finally, I've got some summarize. I have got the conference or hypothesis to use the NanoARC as oxygens for enhanced PDT and boost ROS active for synergistic therapies. And the second, we use the cancer cell membrane and other cell membranes such as the T cell, NK cell, and CAR-T, and for the Biomimetic to realize the homologous-targeting processing phototherapies. And use the PDT initial the immunogenic cell death and boosting our immunotherapies. And also we have tried to fabricate some non-invasive and achieving these devices to get image-guided visualization systems. And also thanks our group members and the collaborations.